Good morning. Good morning, everybody. Welcome to this morning's session on imaging genetics, mapping the effects of genetic and transcriptional variation on the brain. I'm Nicola Palomero, and I'm chairing this session with Casey Paquola, and it's a great pleasure to have you all here. So without much ado, let's get into the first speaker.
Okay. Our first speaker is Adriana Prul, and she'll be talking about alterations of the principal macro-scale functional gradient in carriers of neuropsychiatric CNVs. Thanks, Adriana.
Just make sure it's working. Hello. Okay. So hi, everyone. I'm very excited to be here today. It's a great honor to be part of this oral session. And so, as you can see from the title of my presentation, so I will be talking about the principal macroscale functional gradient and how that is impacted in populations that present a high genetic risk for neuropsychiatric conditions. And I will specifically focus on two of these rare high-risk variants, and that is deletion at the 22q11.2 and 16p11.2 loci, which represent this loss in genetic material on the 22nd and 16th chromosomes, taking out 50 and 29 genes respectively. And the reason why these high-risk variants are special is because they are frequent enough that we can study them individually, and they also present some of the highest known genomic risk factors for a set of neuropsychiatric and neurodevelopmental conditions, including autism and schizophrenia. And here you can see, actually this can give you a sense of their clinical severity. So if we look at 16p and 22q and where they're placed on this graph, we can see that they present much higher risk for autism and schizophrenia compared to common variants, so SNPs, which is the blue cluster that you see on the lower part of the graph. So these high-risk variants enable us to adopt these genetic-first approaches to study brain dimensions underlying neuropsychiatric conditions. And this is what our group has been doing. So one of the strategies that we're using is we're creating these genetically homogeneous groups based on genetic criteria. And in my case, it's based on genetic diagnosis of 16p and 22q deletions. Okay, so prior work from some students in our lab have investigated resting state functional connectivity in relationship to many of these rare high-risk variants. So this is the work led by Moreau and colleagues, and they've shown that there were in fact disconnectivities of in these networks, and these were organized along main functional dimensions. So they showed that there was this interesting pattern of mainly overconnectivity in the somatomotor and thalamus networks, and then in contrast, this interesting pattern of underconnectivity involving the frontoparietal, default mode, and visual networks. And so looking at these results, we hypothesized that there might be a simpler underlying principle for how these rare variants are reorganizing the brain's functional organization. And maybe we're missing it because we're not looking at the right representation of this data. So now introducing gradient connectome analysis. So recent studies have shown this primary gradient in the cortex, in the functional hierarchy of the cortex, that spans from sensory motor to transmotor regions, peaking in the default mode network. And recently in autism, they've shown that this primary gradient of organization was altered, and that it described this compressed cortical gradient with mainly reduced regions in the default mode network. And so in continuity with this work, and also knowing that our deletions increase risk for autism, we hypothesized that this primary sensory motor to transmodal functional gradient would be a key brain dimension between our high-risk deletions, so 16p and 22q, and the neuropsychiatric phenome. So the first aim of our study was to dissect the effects of these high-risk deletions on the sensory motor to transmodal connectivity gradient. So we did that by pooling resting state fMRI data from 2 genetic cohorts, the Simon's VIP consortium and the UCLA data set. This included 19 deletion carriers at the loci 16p and 42 deletion carriers at the loci 22q. We also included controls from each of the scanning sites. So we used this standard pipeline to generate, to preprocess the data and then generate connectomes, and then used a diffusion map embedding to extract our primary gradient, so the sensory motor to transmodal gradient. And then our first analysis consisted of this regional statistical comparison of gradient values. And now I'm going to show the results from this analysis. So we found that there was an altered sensory motor to transmodal hierarchy in our deletion carriers. So if we look at the global histogram that shows the group level gradient values, we find that there is reduced distance between the lower rank regions and the higher ranked regions. So showing that there's some sort of compression here. And then looking at the brain maps that we have here, so these show the FXIs projected back onto the brain. In red, it indicates that there is an increase of these gradient values, and in blue, we find this decrease in gradient values. So what we see is this interesting pattern of compression, showing that lower ranked regions on this gradient tend to have increased values, so they are shifted in the gradient space towards transmodal end of the gradient, and then we find this inverse pattern where the higher ranked regions tend to be shifted towards the unimodal end of the gradient, which is quite interesting, actually. So now we decomposed these FXIs and looked at the proportion of positive versus negative FXIs according to the YEO7 networks. And we find that there is predominantly higher gradient values in sensory motor and dorsal attentional networks, contrasting with lower values in the limbic, frontoparietal, and default mode networks. So one interpretation of that could be that compression of these cortical functional gradient, this particular cortical functional gradient, is in fact one of the underlying principle along which our high-risk deletions and maybe also neuropsychiatric conditions seem to be reorganizing the macro-scale functional hierarchy of the brain. So now for the second aim, we wanted to explore phenotypic associations, so to contextualize these observed differences. So one of the challenges is not so much the pathway from the genes to the brain, but much more the medical interpretation of these neuroimaging patterns. And because we usually have small sample size when studying rare variants, we use this strategy to carry over the deletion profile extracted to the larger UK biobank population that has much more phenotypic annotations available. So this strategy consisted of computing the similarity of each UK biobank subject to our deletion profiles, and then computing this phenom-wide association study with 257 variables available across 10 categories of phenotype encompassing cognitive, mental health, lifestyle, and also physical domains. Okay, so we were able to observe significant association across multiple traits. So as we can see, there are significant associations in lifestyle, mental health, and specifically also in physical and bone density and size, which is interesting. And also we did this testing to see if this was deletion specific, meaning that the changes that are induced by deletions are the ones that are leading to these significant associations. And in fact, we found that random changes of this gradient does not lead to this profile of association, so this phenome-wide profile of associations that we're observing. And this is not so surprising, because we know that 16p and 22q deletions have this multi-system involvement. So carriers tend to have a lot of other health issues, and carriers are usually prone to, may be prone to premature death as well. And finally, we looked at the convergence at the level of the phenome between our distinct deletions. And we find that there is a high convergence, although there remains some specificity. And then something that we would like to investigate in the future is whether these are deletion-specific association profiles or if this is compression-specific. So just to do a quick summary, so we found alterations in our sensory motor to transmodal gradient in carriers of neuropsychiatric CNVs, 22q and 16p. This indicated that there was this global compression pattern. And finally, we also found phenome-wide associations. And one of the future directions could be to extend this to other CNVs and also investigate this compression pattern in relationship to health and disease. So I would like to thank all the people that were involved in this work and also our different funding sources. Thank you.
Thank you, Adriana. Really wonderful work. So we have a few questions already for you. I think we have 3 or 4 minutes as well. So were you able to get any additional genetics of deletion subjects? Did the deletion co-vary with other genetic differences?
Did it co-vary with other genetic differences? Mm-hmm. So this is not something I investigated. So I wouldn't know.
Yeah. No worries. I have a quick question while we get back there. In your gradient study, did you also look at the lower gradients, so the second or the third one that explain less variance?
No, so we really focused on the first gradient for many different reasons, mainly because it's biologically relevant, and also there's this, it's been shown that, it maps this spatiotemporal patterning of the cortical development, and we know that in deletion carriers, there's clearly neurodevelopmental symptoms also that are associated with this genetic conditions. So we decided to focus mainly on this particular gradient, but of course it would be interesting also to look at the second and the third gradient, as they have these different properties. Yeah, thank you.
And time for a last question. So did you take confounds into account using the FIWAs?
Yes, well, so all of the gradient values, so we regressed out the effects of age, site, sex. We also did it with global signal, and we repeated that with different sets of confounding factors also to see how that impacted our analysis. Yeah.
Great. Thank you very much, Adriana.
Thank you.
Okay. And next we have Clara Moreau, and she'll be talking about the genetic heterogeneity shapes brain connectivity in psychiatry. Thanks, Clara.
Perfect. Okay, thank you for inviting me in this session. I'm really surprised to have that amount of people the last day after the club night. So thank you. I was thinking it's going to be empty. So today I'm going to present our last work on the impact of genetic heterogeneity and pleiotropy in psychiatry on brain functional connectivity. So, as you going. Okay, so as you might know, psychiatric conditions have mainly been studied through a top-down approach, starting from the diagnosis, moving down to clinical imaging and genetic dimension. We wanted to identify the gene of autism, the region of autism, or schizophrenia. But so far, researchers mainly reported a lot of heterogeneity at the clinical level with tons of comorbidities. We were not able to find the region or network of autism, and we found instead of that a lot of heterogeneity and overlap between signature. at the imaging level, and at the genetic level, we found also result at the genetic level were mirroring result at the imaging level with a lot of heterogeneity. What I'm saying about when I say heterogeneity, for example, in this last study from our lab in Paris, showed identified 1,500 high confidence NDD genes associated with NDD, neurodevelopmental disorder. And on top of that, 6,000 additional genes were candidate gene also for NDD. This variant were not like on one chromosome, like the chromosome of autism. No, you have they're all over the genome, so it's definitely not easy to study that. And to add complexity on that, we know that a part of this variant associated with autism are also associated with schizophrenia, ADHD, etc. So that phenomena is called pleiotropy. So pleiotropy has been measured using genetic correlation, so SNP-based correlation based on GWAS result. So for example, this last study from the PGC consortium, they correlated results from the GWAS summary from this 8 psychiatric conditions, and they identified 3 groups of interrelated disorders. So for example, ASD with ADHD, as we observed in the clinic, ASD with MD, anorexia with OCD, etc. And a famous one like schizophrenia with bipolar. It's highly related. So at the transcriptomic level, similar approach identified concordant results. So this study, published by Gondal and colleague in Science, reported correlation at the gene expression level across psychiatric condition, and they showed, so in the y-axis, I mean, it's impossible to show you because there's so many screens, but I would like to. But so in the y-axis, and they showed that their result at the gene expression level were correlated with the result reported at the genetic, so GWAS level, SNP-based expression, so x-axis. So we wanted to know if this result at the genetic and transcriptomic level could be mirrored at the connectomic level. So that's the first part. So for this purpose, we used open data set, like publicly available data, so BIDE1, 2, ADHD 200, COBRE, Consortium for Neurophenomics Disorder, and UK Biobank. So we characterized the signature of schizophrenia, bipolar, autism, ADHD condition, control like a non-brain related condition, which is inflammatory bowel disease, and 2 traits on connectivity. We used classical method. We perform connectomal analysis. So we did pre-processing using NIAC software, now using fMRI prep. We parcellated the brain into 64 regions, 12 network, and then we just did mass univariate GLM, and we did simple case control analysis for each of these conditions. And then we correlated our signature of, for example, here bipolar with schizophrenia. I mean, for the one next to me, here and here. And for example, between bipolar and schizophrenia, we obtained a correlation at 0.52. So we reported this correlation in the graph in the middle, and we wanted to know if this correlation at the connectomic level could mirror the result at the genetic and transcriptomic level. So we didn't have a lot of data at the transcriptomic level, but for the few pairs of conditions that we were able to compare, it was quite concordant. So on the y-axis, you have the result at the gene expression level from the Gondal and colleague, and in the x-axis, that's our result. So it looks quite concordant. And then we did the same with results at the SNP-based level, so genetic correlation, RG. So we obtained this genetic correlation from this different paper from the PGC. So for example, you can see on the top, you have this correlation between schizophrenia and bipolar around 0.7 at the genetic level, and we are around 0.50. I showed that earlier, 0.52 at the connectivity level, and for ASD versus schizophrenia, we found something like 0.27, and they found something like 0.3. So it's quite concordant. So we were happy about that. And so in the second part, we wanted to investigate a rare variant, because for now I was only talking about common variant, so SNP-based genetic correlation, so from GWAS. But in that review, we showed that you have a variant, copy number variant, as Andréane presented, associated with ASD specifically, associated with schizophrenia, and that a bunch of them are associated with both conditions at the same time. So we wanted to know if they could impact the same region in the brain, or why, like maybe they are doing like similar things. So in a previous study with Sebastian Erks, in the bottom right on the slide, we compared the impact of two CNVs on connectivity, and we found like similarities in auditory and motor and thalamic area. So we wanted to pursue this work with other genetic variants. So we collected data from several hospitals, including Saint-Justin in Montreal, Lausanne, CHUV, Cardiff Hospital, UCLA, Safari Consortium, because we wanted to characterize the impact of these 15 CNVs on connectivity. So left side, and on the right side, we also wanted to investigate polygenic risk scores, so common variant, SNPs. So that's the agglomerative, the sum of the individual effect of each SNP. So that's how PRS are built. So we were studying EU Biobank, the PRS of polygenic risk score of autism, schizophrenia, bipolar, cross disorder, IQ, and control PRS, not associated with psychiatric condition. So we had like, our hypothesis was the right scenario. Basically, we were thinking that you have more than, as we saw, like we have more than 1,000 genes that confer risk for autism. But at the end, you have like one diagnosis. I mean, you have a lot of heterogeneity at the clinical level, but still you have like a limited amount of diagnoses. So we were thinking that this genetic variant might converge at some place from the genome to the phenome. It might be at the transcriptomic, connectomic, or even like proteomic, metabolomic, blah, blah, blah. We don't know, we don't have those data, but might be that. Otherwise, the alternative scenario is that each gene is going to, in genetic risk, is going to confer like profile, like a clinical profile that is going to be like specific for this gene. So we will have, if we have 1,000 variants, we are going to have 1,000 of clinical profile. So not easy to target, but that might be the reality. We don't know. So that was our hypothesis. So first, we characterize the impact of our genetic risk on connectivity. So on the left side, we only here kept the CNVs with higher effect size, like with connections that survive FDR. Sorry. So as you can see, you have a higher impact for the 22q11.2, but we know that that's a higher risk for schizophrenia, the deletion at the 22q11.2. So that's the highest risk for schizophrenia. And then we wanted to, after characterization, we wanted to correlate the impact of the CNVs with their on connectivity with their impact on IQ. So that's what we represented on the right side of the figure. So on the y-axis, you have their impact on connectivity, and on the x-axis, you have the number of points of IQ associated with each CNVs. So as you can see, you have like a quite good correlation. Then we wanted to search for this hub of like this famous, like we were looking for like convergence, basically. But in the middle, you have like the 12 functional connectivity network. And as you can see, you don't have the region of autism again, or of psychiatric condition. So basically, each network is impacted by this genetic variant, but still we saw like a recurrent overconnectivity of the basal ganglia thalamus and of the somatomotor network across this genetic variant. So we wanted to characterize more formally this observation. So we simply correlated our 20 connectivity profile, like genetic risk, condition, and trait, and we showed like good correlation across these conditions. So we wanted to know which connection in the brain were responsible of this correlation. So we did a PCA across the 2080 connection by 20 FC profile, and we found that connection from the basal ganglia to somatomotor and auditory network were responsible of this correlation. So as a quick illustration, and then I'm done. So for example, if you put a seed in the thalamus, and if you look at the profile of autism, for example, you have an overconnectivity of the thalamus with somatomotor, auditory, and visual network in autism, also reported in schizophrenia in another data set. We found like similarities in deletion 16p in a clinical data set, 22q, and neuroticism. So basically, we are thinking that these similarities of like disconnectivity of the thalamus with sensory motor area could be related to sensory impairment shared across this condition. So it's kind of like we are now thinking about like dimensional approach. Like we are not the only one. So to conclude, we saw that pleiotropy and polygenicity do impact our ability to detect brain connectivity alteration underlying our psychiatric manifestation. We still find, despite this heterogeneity, a critical contribution of the thalamus and the somatomotor networks across our genetic risk and psychiatric condition. And we are thinking that now we will have to cluster rare variants based on relevant gene function. So for example, their involvement at the synaptic level or other like gene function to obtain mechanistically coherent subgroup of individual, because like we will not be able to study 1,000 genes independently, and that we might need to do some partitioning of polygenic score because for now we have a really tiny effect that does not explain that much. So yeah. Thanks to my old lab in Montreal, in particular like Sébastien Jacquemont, Pierre Bellec, Kuldeep Kumar, and Annabelle Harvey, and to my new lab in Paris, so Pasteur Institute.
Fantastic. Thank you so much, Clara. We have time for one quick question. I have one here on the app.
Yeah, I'm sorry.
No, you're fine. Do you have any data on the two different types of 22q deletions and their FC effect size differences?
When you say two different types of 22q?
Yeah. Are there two different types of 22q deletions?
I mean, you can have different coordinates. I don't know. I have two different types. Like you have deletion and duplication, and then you have like different coordinates that like different breakpoint. You have like the big deletion or the small deletion. So yeah, we get data from the small deletion also. Like in UK Biobank, you have some carriers of the small deletion. But I think, if I remember well, we only get like 10 subjects, so it was not enough. Basically, we need like to have like a cutoff of number of like minimum number of subjects we can include, because even if they have like large effect size, like it's why we need like a lot of collaboration and a lot of data from different hospitals.
Yeah. No, it's incredible how many sites you've pulled together. So thank you very much, Clara. Next, we have Elizabeth Levitas. She'll be talking about passing the diversity of gene dosage effects on human brain organization.
Thank you so much. Yeah, my name is Lisa.
Sorry, one second. Could you put that light down slightly, because it's blinding the speakers? Thank you. Sorry.
Cool. All right, I'll start again then. Hi, my name is Lisa, and I'm really excited to be presenting the first project for my PhD, where I've been looking at gene dosage effects on human brain organization and how that relates to regional vulnerability to neurodevelopmental disorders. So I'll start by showing this, which Clara Moreau created and we're all using, and maybe I'm the last one to actually be showing it in this session. But I'll start by situating my project in the context of genotype-first approaches, where we recruit individuals with gene dosage disorders that put them at increased risk for neuropsychiatric and cognitive impairment, and we can measure intermediary brain phenotypes that may potentially relate to these alterations in cognition. And so to date, the bulk of the work in this field has focused on morphometric measures, and so so in this figure on the right, Maldonado and colleagues summarized individual reports of gray matter volume, surface area, and cortical thickness changes in various gene dosage disorders, as well as an idiopathic. How do I use this? Let's see. Idiopathic autism spectrum disorder and schizophrenia. And so obviously there's just an insane wealth of information captured in this with the whole brain patterns across different gene dosage disorders, or GDDs going forward, and imaging-derived phenotypes, or IDPs. And so again, that figure contained whole brain patterns, but I'm going to reduce the schematic down to just 2 dimensions of GDDs and IDPs just to simplify what I'm about to present. And so we can study this in 2 different ways. We can look at multiple IDPs in one GDD, and we can do this in parallel across many GDDs. And Jacob Seidlitz did just that by looking at multiple morphometric measures in individuals who carry an extra X, Y, or 21st chromosome to reveal that integrating spatially dissociable information from these different morphometric measures can reveal unique alterations in interregional morphometric similarity in response to these specific gene dosage changes. An alternative approach is to look at one IDP across multiple GDDs, and this has been done previously in conjunction with voxel-based morphometry to reveal the anterior insula and supplementary motor cortex as having increased or decreased gray matter volume respectively in multiple different copy number variant groups, which are a type of gene dosage disorder. And so we've had these initial cross-disorder and cross-phenotype studies, and my group was motivated to build on these and develop novel analytic approaches to look at many more imaging-derived phenotypes that go above and beyond just morphometric measures, as you'll see in a few slides, and to look at these individual phenotypes that have very distinct genetic influences, as well as gene dosage disorders that obviously have carriage of specific gene changes, and how that may actually shape cortical patterning of regional changes. And so we had 4 specific overarching goals. The first of these was to quantify convergence versus divergence of cortical change across multiple GDDs and imaging phenotypes, to define and compare principal components of multimodal change in these GDDs, to localize and characterize multimodal overlaps in the cortical GDD effects, and to test if cortical regions of shared vulnerability in GDDs also show shared vulnerability in behaviorally defined disorders. And so we tackled this goal by turning to chromosomal aneuploidies, which involve carriage of an entire extra chromosome. And chromosomal aneuploidies have previously been shown to have large effect size changes on both anatomy and behavior. And so we specifically looked at Klinefelter syndrome, or XXY, which involves, in which males have an additional X chromosome, Jacobs syndrome, or XYY, where males have an additional Y chromosome, and finally Down syndrome, where individuals have a 3rd copy of the 21st chromosome. And so across all of these aneuploidies, we had T1-weighted diffusion tensor imaging and resting state fMRI data available. I'll note that all of the individuals had T1-weighted data available, but we had variable case control counts for DTI and resting state fMRI. And after getting all of the neuroimaging data into the brain imaging data structure, or BIDS format, we processed the T1-weighted data using FreeSurfer, DTI using Track2Flow, and resting state fMRI using fMRI Prep and XCP Engine. And so from the 3 modalities, we had, we looked at a total of 15 different phenotypes. And so for each aneuploidy and phenotype separately, we fit a linear regression model to extract standardized beta coefficients for 344 brain regions from the Human Connectome Project parcellation, while also including various modality and dataset-specific covariates. And so this procedure generates a 344 brain region by 15 delta IDP matrix for each of the 3 aneuploidies, which we use to address our 4 goals using the 4 analyses listed over here. And so we can project the standardized beta coefficients onto the cortical surface just to qualitatively assess differences in the brain patterning across the aneuploidies that are listed in the rows and the IDPs that are listed in the columns. And so I'll bring your attention first to a set of morphometric measures that have previously been studied. And so we see that for XXY and XYY, we replicate previous findings of concurrent increases in gray matter volume and surface area in frontotemporal areas, as well as gray matter volume and surface area decreases in occipitoparietal areas. But in addition to those morphometric measures, we also looked at novel phenotypes such as axial diffusivity, which is a measure of microstructural change, as well as amplitude of low frequency fluctuations, which is a measure of altered local neural activity. And so again, just qualitatively, we can see that these maps, A, don't resemble one another, and B, don't resemble the other maps, suggesting that there is a lot of convergence and divergence that we have yet to actually explore. And so we chose to tackle this by turning back to those matrices that are analytical procedure outputs. And so once again, we see that in red, a matrix entry that is colored red indicates that for the specific aneuploidy and IDP that we're looking at, there's an increase in that IDP for cases versus controls. And so we can compile these matrices together to generate this one massive brain region by aneuploidy delta IDP, which is, I know, very wordy, matrix, where we have a total of 45 different aneuploidy delta IDP maps that we're looking at. And so what we wanted to do next was actually compute correlations between every pair of maps in this matrix and perform hierarchical clustering to get at the organizing principles of change. And so here I'm showing that correlation matrix, where an entry that's colored red indicates that there's a positive correlation between two specific maps that we're looking at. Blue indicates that there's a negative correlation, and where you see an asterisk, that means that there's still a significant correlation after performing a spin test. And so we also color-coded the individual maps based on the aneuploidy that they correspond to, as well as the modality that they correspond to. And so this revealed a first striking principle of organization, where we have this major cross modality split with more subtle subdivisions across different phenotypic groupings, such as diffusivity measures versus anisotropy measures. And then within that, we have splits across the different aneuploidies that we're considering. And so we also sought to represent this information using a force-directed graph, which, for the sake of time, I'll just say this just like further drives home the split that we're seeing in the correlation matrix, with T1 weighted measures on one end, DTI measures on the other end, and then within those, we have a split across the aneuploidies. And so this first analysis allows us to actually look at gross convergence and divergence across all of the maps that we're considering. But what we wanted to do is go back to those whole brain patterns that we were seeing in the Maldonado figure and actually understand where is this regional brain variation stored and derive the primary spatial axis of multimodal change. And so once again, we turn back to those three matrices I had shown you earlier. We apply principal component analysis separately to each of these. And on the right, I'm showing a scree plot with the variance explained for the first 10 principal components for each aneuploidy. I'm going to be focusing on just the first principal component going forward. And for XXY, XYY, and Down syndrome respectively, the first PC explains 38, 31, and 25% of the variance. And so the PCA returns two major features of interest that we can explore. The first of these are PC1 scores, which effectively reflect which regions are most similarly affected across all of the phenotypes that we're considering. And so we see, for example, for XXY, we have this anterior to posterior gradation. We can compare the PC1 scores across the aneuploidies, and we see that for XXY versus XYY, we get the highest correlation of 0.73, with considerably lower correlations between Down syndrome and the sex chromosome aneuploidies. We can also look at the actual imaging-derived phenotype loadings onto this first PC to actually understand what the phenotypic contributions onto this primary spatial axis of multimodal change is. And just briefly, we see that for XXY and XYY in pink, we have counterpoised contributions between morphometric measures all the way on the left and diffusivity measures on the right. For Down syndrome, we're seeing counterpoised contributions between anisotropy over here in red and the morphometric measures in red on the left. And once again, we do get a much higher correlation between the phenotypic loadings for XXY and XYY with a correlation of 0.95 and variable correlations for the other aneuploidies. To actually provide a richer biological annotation for these PC1 maps, we performed a spatial comparison with a canonical resting state parcellation, specifically the Yolk-Renin 17. And just briefly, we mapped brain regions from the HCP parcellation onto the Yolk-Renin 17 subnetworks, computed the average PC1 score within each of the subnetworks, and performed a spin test to actually assess the significance of the enrichment. And so we find that for XXY, low PC1 scores are significantly enriched in a somatomotor subnetwork. For XYY, we have significant enrichment of low PC1 scores in a visual subnetwork. In Down syndrome, we have many more clearly significant enrichments, one of these being high PC1 scores are significantly enriched in a limbic subnetwork. And so finally, this brought us to our last analysis, which is, can we actually localize overlaps across these principal components for the three aneuploidies to understand which brain regions are convergently affected across many different modalities within these aneuploidies? And so for this, we extract the low and high deciles for each of the vectorized PC1 scores, and these are in blue and red, as well as the middle decile, which is how we refer to sort of the neutral brain regions that we consider to be resilient to gene dosage change. And so when we do that, we can actually unveil two common patterns of brain vulnerability in blue and red. And so in blue, we see the superior parietal cortex and bilateral visual areas as those that are convergently vulnerable on the low end of the PC1 map, as well as the left middle temporal gyrus and the right insula for the high PC1 scores. And so when we pause and actually look at this, what was particularly interesting is that the right insula and the insular cortex in general is a brain region that's been time and time again been shown to have convergently reduced gray matter volume across a number of different behaviorally defined psychiatric disorders. And so this brought us to our final question. If we were to actually get at the primary spatial axis of multimodal change in behaviorally defined psychiatric disorders, would we see alignment between that and the average PC1 maps that we have for the aneuploidies over here? And so for that, we turned to the really just wonderful Enigma toolbox, from which we got 46 case control maps for 5 different behaviorally defined psychiatric disorders: ASD, ADHD, schizophrenia, bipolar disorder, and epilepsy. They had data for cortical thickness and surface area, and we once again just applied a principal component analysis to get the first PC reflecting the primary spatial axis of change. And I'll note again, actually, that these ones are provided for free surfer regions, which we just map onto the cortical surface. And if you recall, I was previously working in the space of the HCP parcellation. So after remapping the PC1 scores that we had onto the Desikan-Kiliani regions, or the free surfer regions, we're able to actually compare our maps and find a really strong correlation of 0.69, which remains significant after performing a spin test. And so, in summary, our study provides the first deep phenotypic analysis of multiple chromosomal aneuploidies. We find that multimodal changes are organized first by phenotype, with subtle divisions across aneuploidies. Principal components show variable correlations with respect to both regional scores and phenotypic weights, reinforcing genotype-specific nature of the multimodal effects. And finally, overlapping changes in aneuploidies echo overlapping changes in idiopathically defined disorders, or behaviorally defined, suggesting that there are certain regions that show recurrent alterations in neuropsychiatrically relevant disorders, defined either top-down or bottom-up. With that, I'd like to thank my supervisors at the NIMH and UCL, who are Armin Rasnahan and Daniel Alexander, as well as all of my collaborators, the families who have participated in these studies, and thanks to all of you for your attention.
Thank you, Lisa. Okay. Well, by the way, I'll remind everyone, if you would like to ask a question in person, I can also pass you a microphone if you want to do it old school. So we have a few questions from the app, though. So, here's one. Can you speculate about the causal mechanisms underlying all these genome-brain-phenome association patterns?
Yeah, so one thing that we actually started doing, and I haven't included in this presentation yet as it was all very preliminary. Oh, sorry about that. We've started actually trying to incorporate an analysis using the Allen Human Brain Atlas to get at potential molecular underpinnings that are shared across all of these different disorders. But can't really speculate too much yet because those were super preliminary results.
That sounds really exciting. Maybe one more quick question. So how do you interpret gene morphology association in a cross-sectional study? So is it possibly some inborn traits from some risk allele carriers?
That's interesting, yeah. And I mean, one thing that my lab has discussed a lot is that there is definitely a strong need to incorporate longitudinal data, which unfortunately we don't have at the moment for, 3T level data. But sorry, can you repeat the first part of the question?
No, I think you answered it. I think, okay. Yeah, you're quite aware that longitudinal data will help you map those.
Yeah, and I guess I'll also note as well that unfortunately, if you recall from the the demographics table that we did have reduced sample sizes for specific modalities. And so unfortunately, there is just reduced power to detect some of the effects.
So, any other questions? I think that's great. Thank you so much. Okay, next we have Janine Beisterbosch, I'm sorry, and she'll be presenting on behalf of her postdoc, Wei Zhang, on shared brain and genetic architectures between mental health and physical activity. Thanks, Janine.
Thank you so much. Thanks for inviting me and for coming today. Yeah, unfortunately, my postdoc couldn't be here with you all, and so I'm honored to present her work on her behalf today, looking at the neural and genetic architectures that are shared between mental health and physical activity. So the idea behind this project was that both mental health and physical activity impact our emotions, impact our cognitive abilities. A lot of work has shown that doing some physical exercise has a positive impact on psychiatric illnesses such as depression, and is suggested as a good way of self-care nowadays. And so we really wanted to look whether there was, given all of this known kind of linkage between these two phenotypes, whether there was a shared basis both in the neural architecture and in the genetic basis. And so in terms of describing the methods on the imaging side and looking at the shared brain architecture, we used for this data from the UK Biobank, about 6,000 subjects who had data on all of the different measures that we're using. We wanted to focus on some of the more higher order cognitive networks. So we're using resting state data from 7 subnetworks defined using independent components analysis that together comprise the default mode network, the salience network, and the central executive network. And so our features on the imaging side are 21 pairwise connections between those 7 subnetworks, kind of always, and the 7 mean amplitudes or strengths. And then on the phenotypic side, in terms of mental health, we use the self-reported questionnaires in the UK Biobank, which has 31 questions, and we performed a PCA to remove some of that redundancy across those questions, retaining the 7 PCs that together explained at least 50% of variance. And then lastly, on the physical activity side, we use data from an accelerometer, a wrist-worn sort of step counter that people were given to wear for, I think, a week or two. And a previous study developed a machine learning approach to extract summary measures from that. So we had summary measures on the duration of light activity, moderate activity, sedentary activity, sleep, things like that. And so the approach that we took, and then we had some covariates that I'm listing here. The approach that we took was to use a canonical correlation analysis, and we performed separate models independently relating mental health to the brain measures, and then relating physical activity to the same brain measures. And then in addition to that, we also did what we're calling unique canonical correlation analysis models, which are the same but essentially covariating out the other phenotypes. So when we're looking at the relationship between mental health and brain measures, we're controlling or treating the physical activity measures as covariates, and vice versa. When we're looking at the relationship between brain measures and physical activity, we're treating the mental health measures as covariates. Moving on to the genetics, so here we use summary statistics from previous GWAS studies. Those summary statistics were available for the same identical physical activity phenotypes. They were not available for the identical mental health phenotypes that we used, and so on that side we used GWAS summary statistics that overlapped conceptually with the mental health areas that we were looking at. So the list that you see here kind of captures all of the self-report questions that were in the questionnaire that we covered. And so those were used, first of all, to do a pairwise linkage disequilibrium score regression to look at essentially genetic correlations separately within each construct, so amongst the mental health phenotypes and amongst the physical activity phenotypes. And then we performed genomic structural equation modeling to identify a latent factor of mental health and relate that in terms of its genetic correlations with physical activity measures. And so time for some results. This is a good one for anyone who was at the club night yesterday, because this one is interpretable with a fuzzy brain by just squinting a little bit. These are some results from the canonical correlation analysis. And so what I'm showing you here are loadings, so essentially feature contributions of all of the brain measures that we had for two separate analyses. So on the left-hand side is mental health, and on the right-hand side is physical activity. And like I said, just squinting, you can see these patterns look very similar. I'll draw your attention to a few more things here. So the top list of the 21 measures at the top, those are the connections, the pairwise edges between the brain subnetworks that we looked at, and the bottom ones in orange are the strengths or the amplitudes. And so I'll draw your attention, for those who are a little bit more awake, to some specifically interesting things. So it was the dorsal default mode network and the left central executive network that were contributed the strongest, both in terms of their amplitude and the connection between those. And another result that we found surprising is that these amplitudes at the bottom consistently showed the highest loading. So they actually contributed slightly stronger to this result than any of the connectivity measures. And now I'm going to switch to the next slide, and I'm going to change these figures at the bottom to the results that we got when we controlled for the other phenotypes. So doing the mental health controlling for physical activity, and doing the physical activity controlling for mental health. And you got to pay attention, because they're almost identical. And so what that shows us is that, first of all, because these patterns look so similar, it shows us that these two different phenotypes of mental health and physical activity really do map onto very similar kind of brain bases. But because the unique results don't change very much, they kind of have an independent mapping. They don't depend on one another. It's not like they're causally related to each other. These are some results for the pairwise genetic correlations within each of the constructs. And so I think there's no big surprises here. You see relatively high genetic correlations for phenotypes that are similar, and you see negative correlations for example, looking at sleep and sedentary behavior related to when you compare that against more active exercise type behaviors. And so my last results slide is to show you the results of the shared genetic architecture. So here, first of all, we identified a kind of latent factor of mental health that we're calling negative affect, that had pretty strong contributions across a number of things. And that was significantly genetically correlated with a number of our physical exercise measures, including moderate activity, overall activity, and sleep. When we control for body mass index, some of those effects go away, so the effect of sleep stays, but the effect of overall and moderate activity do not survive controlling for body mass index, which makes sense because body mass index is closely related to how much activity you would have on average. And so in summary, hopefully I've shown you that we have evidence for a shared neural architecture between the phenotypes of mental health and physical activity in these kind of higher order cognitive regions, such as the default mode network and the central executive network in particular. I've also shown you that there's some evidence for a shared genetic basis, particularly driven by sleep and some moderate and overall activity. So altogether, this points to a potential mechanism that might explain some of the positive impacts that we see from physical activity onto mental health. And so with that, I'll say a really big thank you to our collaborators, our funding, my entire lab at Washington University in St. Louis, and in particular to my postdoc, Dr. Wei Zheng, who really spearheaded all of this work, and she's with us in spirit today. Thank you very much.
Thank you so much, Janine. That was wonderful. And thanks for calming our fuzzy heads after club night. So we have a question here. How do you account for the long time gap between questionnaires and the date of scan?
Oh, so I'm trying to think, and this is my fuzzy club night brain. For a lot of the studies that we do, so there's two different mental health measures in the UK Biobank. There's some stuff that gets done on an online questionnaire and gets sent out, and there's a really long time gap. But there's also some mental health measures that are done on the day of scanning. And for a lot of the studies in our lab, we focus more on the ones that are done on the day of scanning. And I want to say that we did that here as well, but I'm not 100% sure.
That's fair. Thank you. And another question. So which causal direction do you think is more plausible?
Oof, causality is a scary word at any scientific conference, I feel. I think the association with sleep is interesting, and so which is in itself causally difficult to interpret because sleep is both sleep can cause worse mental health, but is also a symptom of worse mental health. So that illustrates the challenge and my nervousness using the word cause. And so I think it probably goes both ways. I like the physical exercise as a kind of self-care approach, because I think once people get really depressed, it's hard, even if they cognitively know, like, this is going to help me if I go for a walk or a run or something. The symptoms of depression prevent them from having that motivation to be able to do that. And so I think it's really multi-directional depending on where you are on that continuum of mental health symptoms.
Yeah, it really speaks to the vicious cycle that you have between these aspects.
Yeah.
I think that's, we'll move on to the next speaker. So thank you so much, Janine.
Thank you.
And next we have Richard Deere. He'll be speaking about 3 principal components of human cortical gene expression.
Okay. This one. Great. Oops. Okay. Hi. Good morning, everyone. My name's Richard. I'm supervised by Petra Berters and Ed Bormore at the University of Cambridge, and I'm really excited to talk to you about these 3 gradients of gene expression. So, in the last decade, there's been a whole lot of really exciting work using the Allen Human Brain Atlas, which measures gene expression at about 2,000 points across the cortex, to decode brain maps in terms of genes. And that gives rise to the question of whether the brain's gene expression might have its own intrinsic structure independent of any of these specific maps that we compare it to. Now, I'm not the first to ask this question. A few years ago, Josh Burt showed that if you just take the Allen Brain Atlas and put it into a parcellation so that you have a matrix of, say, 180 brain regions by 20,000 genes across the top, then the first principal component of this matrix is this smooth gradient across the brain. And that's without having any other spatial information in the matrix. Moreover, this first component is very closely aligned to the MRI-derived T1-T2 ratio, which indexes gray matter myelination as well as cortical hierarchy overall. Now, Valerie Sidnor and others had an amazing review where she showed that this concept of hierarchy extends to a whole range of canonical brain maps that all have some alignment to this sensory-motor association axis that in some sense is underpinned by this first principal component of gene expression, which is this one. And that made me really curious about whether all these imaging maps and all the other gradients we see out there in the world are all organized around just this one single gene expression axis, or whether they might be underpinned by multiple intersecting axes of gene expression that organize all these 20,000 genes, which is another way of saying, what about gene expression PC2 and PC3? Now, it turns out if you just throw the Allen Brain Atlas into PCA naively, it doesn't look like there's anything more than PC1, because PC2 and PC3 just look very noisy. But what I'm about to show you, this is the teaser, is that if you clean up the data in a bunch of ways, you can find these 3 smooth gradients that are biologically meaningful and relevant. So the key challenge to doing this is that just because we can compute higher order principal components doesn't mean that they're real. I mean, we can compute PC100 if we like, but all of these data come from just 6 brains in the Allen Brain Atlas, so at some point, the higher order components are just going to be overfitting to the specific individuals in the data set. So how do we know how many principal components are real? In particular, this is the only data set that we have that has high spatial resolution of gene expression. So our solution to that was to make the problem even harder. We split the 6 brains into triplets, process them completely independently, and see if we can find the same gradients from these different independent data sets of 3 brains that have no brains in common. And if we consistently are finding the same gradients when we do that between every way of splitting the brains into disjoint triplets, it suggests that if I were to take 3 of your brains and measure the gene expression non-invasively, then we would also get the same result. In other words, that it's generalizable. So here's the results of this when we apply it to all the data. Each point here is the absolute correlation between one way of splitting the brains into triplets for the 3 components. And you can see that PC1 is pretty consistent, and PC2 and PC3 look like rubbish, which makes sense because they're so noisy. However, other studies have shown that not all the genes in the Allen Brain Atlas are equally stable. Here I've got the same data with the 3 principal components plotted on top of each other. And on the x-axis, I've instead got how many genes we're retaining after filtering the genes for just those genes that are more stable. And we do that. That just means how similar is the brain map of the individual gene between each pair of brains in the triplet in this case. And you can see that the intertriplet correlation increases as we filter for only the more stable genes. But we want to retain as many genes as possible to have the most biologically interesting result. So can we do any better than this? Well, another reason the data is very noisy is because not every region has samples from all the 6 donors. 43 of the 180 Glasser parcellation regions only sample from 1 or 2 of the donors. And that means PCA isn't going to be able to distinguish the common spatial variation from what's specific to those donors. So let's just drop those brains. If we drop those regions, sorry. If we filter out those 43 regions, we have a few gaps, but this intertriplet correlation becomes much smoother. And say we set a threshold of we want the intertriplet correlation to be at least 0.6. Now we can include 20% of genes and have all 3 of the gradients meet this criterion. And we're starting to get something that looks a bit interesting here. But wait, if we change from PCA to diffusion map embedding, everything looks better still. And we can perform the same analysis, and now it looks like we can go to 50% of genes, that's about 8,000 genes, and still meet that criterion. And the exciting thing about diffusion map embedding is if we now relax that region filter, so we don't want these patches missing in the brain, yes, it gets worse, but not that much worse. We can now go to 20% of genes and still meet that threshold, which gives us these really lovely smooth brain maps that really look like something that may be interesting. So are they interesting? Well, the first way to look at that is to take gene weights from the brain maps. Here I'm using the ones that have 50% of genes. And the one thing I forgot to say is that all these sets of maps with different parameters are very highly correlated with each other. It's about 0.9. So it's just a different trade-off between whether we filter genes or regions to get the same result. And when you look at the gene weights from those maps, we find that the second gradient is very strongly enriched for metabolic processes in the positive end and epigenetics at the other. And the third gradient is even more strongly enriched for inflammation at the positive end and synapse development at the other. And that aligns with cell type enrichments, where we see, for example, that all the glial cells are super strongly enriched in the inflammatory end of the third gradient and negatively enriched for synapses and neurons at the other end. So now turning back to all the interesting canonical brain maps, these gradients that we see in different papers in the literature, one really interesting result is that all these maps are somewhat correlated to the first gradient, but actually half of them are more correlated to the second metabolic gradient of gene expression than they are to the first myelination gradient. In particular, look at aerobic glycolysis, which has almost a 0.7 correlation with the second gradient of gene expression. That makes sense given the metabolic enrichment. Even more interesting, if you just take this set of brain maps, which don't have gene expression in them, or any subset of them, then the first principal component of these canonical imaging maps is actually the second metabolic principal component of gene expression. And vice versa, the second principal component of the imaging maps is the first component of gene expression. So it really seems like these canonical imaging maps are underpinned by multiple axes of gene expression in the brain. Now, you may be wondering, what about the third inflammatory gradient? That doesn't seem associated with any of these. That's true. But we also had a disorder data set, which Richard Bethlehem and others put together from a huge sample of nearly 150,000 MRI scans. So these are cortical thinning from normative models of autism spectrum, major depressive disorder, and schizophrenia. And we see that when we associate those with these gradients, ASD is very strongly associated with the first and second gradient, and schizophrenia is specifically associated with the third gradient. These are all spin tested and multiple comparisons corrected. And as usual, MDD doesn't have much correlations with anything, sadly. But this is particularly interesting because ASD has some links to metabolic dysfunction and SCZ has some links to inflammation. And it also suggests that maybe there's something going on with neurodevelopment here. So to look at that, the final analysis was to say, what if we take a developmental data set, BrainSpan, which also measures gene expression only consistently in about 11 regions, but at brains from different ages of development, and map those regions to the Glasser Atlas, then take the gene weights from our gradients and see if we get the same spatial pattern in those 11 regions from these two different data sets. And so that's what we're doing here with just the adult brains, and we see that all three gradients are very strongly aligned over those 11 regions in the adult brains from BrainSpan. So that's a nice validation. But even more interestingly, if we do that same analysis at earlier ages in development, we see that the first two gradients emerge early in development, in that they're quite consistent with the BrainSpan data, even for prenatal brains. But the third gradient seems to only become consistent, only seems to emerge after adolescence. And that aligns with autism spectrum being a disorder that we can diagnose early in development, but schizophrenia being something that where the symptoms only emerge around adolescence. So altogether, I've shown that if you process the Allen Human Brain Atlas with some gene and region filters, it reveals 3 robust gradients of gene expression in the brain that seem like they might generalize. The second gradient is metabolic, prenatal, and links to ASD, while the third gradient is immune, post-adolescent, and links to schizophrenia. And altogether, that suggests that the anatomy, development, and disorders of the human cortex are likely to be underpinned by multiple gene expression gradients, and possibly even more if we had more brains one day from the Allen Atlas. So with that, I'd like to say thanks to my amazing supervisor for this project, Petra Vertes, all our collaborators that made this possible, my funders that mean I can be here, and to all of you for coming along after club night.
Thank you so much, Richard. Wonderful. Okay. Are there any questions in the crowd? If not, I'll go. So how do you make sure that you're not fishing when you're doing these reliability tests with all the changes in the percentages and the way to select genes? How do you make sure that doesn't become a fishing expedition? Someone is asking.
Yeah, I think one answer is, in a sense, it is. But another way to think about it is that this triplet way of splitting the brains into triplets is a metric for how to optimize the processing parameters of how we look at the data set. I mean, there are so many ways and choices that we have when we process a data set with any data set that we look at, particularly something that's high dimensional and complex. So it's very useful to have a way of saying, what are the parameters that are best extracting the signal from the noise? And I think that's the way to understand what we're doing here.
That's very nice. I've got a really stupid question, and I'm going to blame it on the Club Night fuzzy brain. You said that you started off with the 6 Allen brains,
Yeah.
and you created triplets. So
What
I
does
was
that
trying
mean?
to, I was trying to get my fuzzy brain around that, and it's embarrassing. You know, I know what a PCA is, and I don't know what.
Sure, no worries. So here we have 6 brains in the Allen Atlas, and so you can split the data. So you have 3 brains and 3 brains in the other. So the data comes as a gene expression data set for each brain individually. So I'm just saying, let's take 3 of those brains and then the other 3 of those brains and process them independently, and then compare them and see if we get the same result, and then do that for every way of splitting the brains.
I understand. Okay.
Yeah.
I also have a question.
Yeah.
Why do you think diffusion map embedding is superior to PCA?
Yeah, well, I think because it's a nonlinear dimension reduction technique, and there's no obvious reason why all these 20,000 genes should organize around linear combinations in the brain where they just add up together to form a single gradient. I mean, diffusion map embedding is really just asking, can I find orthogonal spatial patterns? So these are still orthogonal with each other spatially, but it doesn't also require that the gene weights are orthogonal in the way that PCA does, and that's probably more biologically realistic.
Great answer. Thank you.
Thank you.
Yeah, thanks.
Was there one question? We can have one question if you like. I love in-person questions.
Yes.
Oh, thank you.
I was wondering whether you tested out different postulation schemes.
Oh,
So the question was, just to repeat it into the microphone, is did you test out other postulation schemes?
can I have my slides back? I have a backup for that. Maybe not. Maybe that's too. The answer is yes. So you get the same result with the gene weights being correlated above 0.9 for each gradient in Descon-Kiliani, Destereo, Schäfer 400, and they all correlate with Glasser. But Glasser looks the best by far.
A strong yes. Thanks, Richard. Okay, and next up we have Jake Vogel talking about whole brain transcriptomic axes of human brain organization. Thanks, Jake.
All right, thanks so much to the organization for the opportunity to give this talk about the real gradients, Richard. No, I'm just kidding around. But actually, I think one important distinction between what I'm going to show you and the fabulous work you just saw was that we are using sort of the entire brain to look at this question rather than just the cerebral cortex. So I'm going to start with a very familiar picture here that you all know of the brain. And I think this picture illustrates something that I really used to take for granted a lot, which is the fact that function is distributed in space in the brain. You think that's pretty obvious, but there are organs in which that's not the case. Like if you think like the epidermis, the skin, this is an organ in which function is pretty much uniformly distributed. In the brain, that's not the case. We have cell populations. We have nuclei that are participating differentially in behavior. And that's something that's probably sort of consolidated and caused by some early developmental processes that occur in regionalization, arealization process I'll sort of walk you through that are actually sort of controlled by brain gradients. So I'm going to do that with this really janky cartoon I made. So imagine you've got a neural compartment. Maybe it's the spinal cord or perhaps the cerebral cortex or something. In the developing brain, you have a patterning center, and you have morphogens that sort of diffuse out into this compartment, creating a concentration gradient. And that will attract different proteins to different parts of the compartment based on their differential repulsion or attraction to that signal. Now, let's say one of those itself happens to be some sort of molecular source that's itself diffusing out into this compartment, causing a more complex gradient. And this, again, causes more proteins to go into different parts of the compartment. And eventually, you get this mosaic of different areas distributed across this compartment that have distinct molecular features, and over the course of development, become functionally distinct as well. So this is very well described and very well studied in the model animals. This is just an example from the spinal cord. Same idea: diffusion of morphogens into a compartment, differential recruitment of different proteins causing different areas. And the question is, and so I think something this teaches us is that with just a few molecular gradients, you can have pretty substantial regional diversity. And the other thing that's really interesting is it really enforces the principle that different parts, that function gets distributed in space. Like it's sort of almost a requirement. So the question is, how does this relate to cortical topography in humans? We do know that there are proteins that are expressed in a gradient-like pattern in the human brain, and we even know that there are some theorized whole brain gradients that sort of where you have gene expression or expression of a protein across the cardinal directions of developing brain. I'm kind of highlighting this a little bit better here because that's a black and white photo or figure, but you can see that you have these vertical gradients that stretch across singular compartments, as well as longitudinal gradients that go across the entire developing brain, and some sort of horizontal gradients here that are really mostly in the cortex. And so this is pretty hard to measure. You need really high spatial sampling of human tissue. And so, well, we do know that we have the Allen Brain Atlas, which I won't go through. I think you all know pretty well. But it does allow us to ask the question whether these sort of, orthogonal directional gradients that we see in early brain development are in fact neotenous features that we can maybe detect in the adult brain, because obviously these are adult brains in the Allen Brain Atlas. So that's the question we move forward with. And the way that we sort of address this is using cross-decomposition. So you probably know PLS or CCA. We use PLS here, partial least squares regression, where you basically have two different data sets and you want to find latent covariance between those data sets. And in this case, the two data sets are on the one side, gene expression across different tissue samples, and on the other side, literally the spatial location of those same tissue samples. And what we get out of that, the components that come out of this, are just directional autocorrelation of gene expression. And we can ask, essentially, what are the primary directions across which gene expression is varying in the brain? So I'm going to sort of omit some methodological details, which I'd be happy to talk to anybody about later. But in the end, we end up with these three components, significant components. On the x-axis of each of these is a latent gene expression component. On the y-axis is a latent spatial component. And together, they make the sort of covariance components of the PLS. And I'm just going to display these on the entire brain. You can see in each of these maps, it's all of the tissue samples in the Allen Human Brain Atlas, specifically the Y and Z coordinates. And in the middle, you see these spatial gradients. It's literally just the direction by which gene expression is varying. And on the right side, you have these transcriptomic variants, which is literally gene expression that is varying along those directions that are enforced by this sort of first spatial component. And we were really interested to see this because it looked a lot like these theorized sort of developmental whole brain gradients, right? So for example, you have this sort of hindbrain to forebrain radiation, which is really similar to what we see in the first one. The second one, again, you've got this anterior, posterior radiation that, again, happens throughout all the compartments of the brain. And then the third one, we even get this kind of sort of diagonal, medial lateral, dorsomedial to ventrolateral sort of directional radiation, which was really fascinating to see because it's purely a data-driven approach. So the question that we sort of started with is if you sort of bring these gradients together and let them interact, these whole brain gradients, in the cerebral cortex, do you get recognizable functional divisions, recognizable brain regions? And so we took each one of those three gradients and we projected it onto the cortical surface. We did a clustering analysis to just find a sort of unsupervised territories that have sort of different concentration of these various gradients. And for the first time in my life, every clustering metric I used suggested the same number of clusters, and that was 6. And so when we looked at those clusters, they just kind of looked like the cytoarchitectonic divisions of the brain, the almost lobar structures that we all recognize. We were planning to run a neurosynth analysis, but it's like there's no point. Like we know what those regions do, right? It's just, so that was really cool to see. And it also, I think, emphasizes the point that these like large lobar structures are actually sort of distributed orthogonally in space. Like we don't think about that. It's like the frontal lobe because it's in front. I mean, yeah, anyway. So, right, so you already, Richard already brought this paper up, but I do want to emphasize it again. SAR grad student in the Satterthwaite lab, Val Sidner, had this really wonderful review I suggest you all read, talking about the sensory-motor association axis, otherwise called the hierarchical functional hierarchy or the principal functional gradient or what have you. And it really does seem like, from this review, a backbone along which so many different cortical features vary. And so we decided it would be interesting to compare our sort of 3 molecular gradients to this primary functional gradient in order to see which sort of best explains the topography of these several different brain features. And so to be fair, we actually tested them against the top 3 functional gradients. And we just fixed linear models with one set or the other set and asked which one of these models explained the cortical topography of these features better. And we found that across nearly all features, the molecular gradients better explained the topography of these cortical features. So that was pretty interesting. And then we were curious, well, okay, so this seems to be important for creating these functional divisions, but does it end there, or is there some link to brain function in the adult brain? Is there sort of a gradient-like representation? So the first thing we thought about was these traveling waves, which really represents local field potentials oscillating in different wavelengths along the cerebral cortex. You can measure that with ECOG. We did not have ECOG data, but we did have these MEG maps that lets us theorize this sort of local field potential, or estimate these local field potentials. And so we did that, and so we sort of looked at the sort of MEG patterns of the diffusing local field potentials across different wavelengths, and we saw that different wavelengths corresponded to different molecular components, right? So theta waves in the case of C1, alpha in the case of C2, and gamma a little bit more weakly in C3, but all of them were really strong sort of directional correlations in space and all survived spin tests. So to wrap this up, we were finally curious how consistent these were. If these were really sort of organizational structures for development, we should see them across probably primate species, across development. In order to do that, we needed to make sure that they replicated in other data sets. This is something very similar to what we just saw Richard present, where essentially we have our PLS model that was sort of trained in the discovery data set. We use that to transform different brain regions in our discovery data set and transform the same regions in the external data set, and then we simply correlate them to make sure they're similar. And when we do that across 3 different other data sets, BrainSpan, GTEx, and PsycEncode, we see very strong sort of reproducibility of each of these components, which just means that the gene expression patterns across regions are very similar, so we can make apples-to-apples comparisons. And so this can also be done at the individual level. You can compare a single individual to the expected sort of discovery data set regional pattern. So we did that. What you see here, each one of these dots represents a different individual. The y-axis here is regional correlation to the discovery data set. And you can see across human, macaque, and chimpanzee brains, we have really consistent reproducibility of the first 2 components. But the third component, we did see a component by species interaction whereby the different primate species had a lower reproducibility. Interesting also because this was the gradient where we had the most cortical variation. And finally, we did the same thing across development as well. Again, each dot is an individual. X-axis this time is sort of log age from prenatal all the way to adulthood, and y-axis is similarity to the discovery data set, which is adulthood. So it's similarity to the adult gradients. And what we found is that across 2 different human data sets and even across 1 macaque data set, we had very consistent sort of developmental timelines, which were different for each gradient. But also interestingly that they all sort of converge in adulthood, suggesting that perhaps these are residual or sequelae of the original gradients. Yeah, so a lot of interesting stuff in there. So I just threw a lot of stuff at you. I would be very interested in your thoughts. My sort of takeaway from this is that, these are actually really basal organizational scaffolding of the brain and of the cortex, and things like the functional hierarchy probably sort of emergent to optimize the sort of communication between these distal sort of in very different regions. And I'd be interested in your thoughts, but first I'd like to just thank all the collaborators that participated in this, the Open Data Sets and the Satterthwaite Lab for sort of all their help with this, and especially Jacob Seidlitz for his really crucial contributions to this project. Thanks.
Thanks, Jake. Awesome stuff. Okay, we have a few questions here. I'll start with one that I think it's just a clarifying question for someone. They asked, did you look at subcortex?
Yeah. So we didn't use a parcellation at all. We just took every tissue sample across the entire data set. So that includes cerebellum, subcortex, brainstem, midbrain, the whole 9 yards.
Nice. And this builds on that, actually. So do you think we can parcellate the brain by using the molecular gradients, e.g. by doing an ICA to further parcellate?
Yeah, definitely. I think if I go back to like the original, I guess you could just see it here. If I look at this at another sort of resolution, there were other sort of like peaks further along at like 13 regions, and when you do that, you do again see these very interesting and recognizable structures. And I think that it can probably also be used to do the same thing, not in the cortex, but in the rest of the brain, perhaps to see if there are some interesting divisions there. So it's certainly possible.
Maybe I have a little follow-up to that. Are there parts in your genomic, you call them gradients,
yeah,
but are there ever sharp changes?
By design, no, because the way that we sort of created these, well, actually, you can sort of see here.
Yeah, did you smooth the data beforehand?
No, the data was not smooth. It's just sort of, but space is sort of like, in and of itself, kind of smoothing what's going on. So that is for sure a limitation in this. But the reason we did this is that even though we do see sharp divisions later on in development, the original sort of radiation of these morphogens is sort of just a diffusion through space. And that's sort of what we were kind of after. But that's a great question.
Okay, and this is like a kind of a speculative one. So what are your thoughts on why these presumably early developmental pattern gradients are preserved in expression in data much older, such as the adults in the Allen Brain Atlas?
Why are they preserved? Yeah, so that's something that I've been curious about myself, and probably just because they are part of the scaffolding of the construction of the structure of the brain. But I don't think it stops there. I think that there probably is, we see autocorrelated variation in lots of different measures, not just with imaging, but actually in the biological measures themselves. And so I think that we should continue to actually really explore this, because I think it's more basal. Maybe it's not very human specific, which is what we're often interested in, but I think that we should continue to characterize what's going on here in sort of along these very cardinal gradients in the brain. Because every time we look at the gene expression data, like those are, I think, the main sort of sources of variation that we see.
Yeah. That's fantastic. Thank you so much, Jake.
Thank you.
Okay, so thank all the speakers for their wonderful presentations, and of course the audience for listening and for the interesting questions. And with this, we'd like to close today's session. Enjoy the rest of the conference.